Now we are going to talk about RFLPs. People don't use them as much as what they used to use them before because now we have PCR technology. However, it is important that students know about RFLPs because they will invariably encounter this. So let's look at this. RFLPs basically means restriction, fragment, length, polymorphism. Restriction, of course, means the fragment that is generated by an endonuclease DNA fragment. So R is restriction. I have explained what that is. Fragment, I have explained what that is. Length, polymorphism. So length polymorphism is basically means more than one shapes or more than one forms. So because when mutations or changes occur in DNA, they can actually add or remove restriction sites or those palindromic sites where restriction enzymes can cut. So here, let's do this exercise. Here we have a DNA sample. We use this DNA sample and we cut it with an enzyme 1. We cut the same fragment with enzyme 2 and 1 together. So when we run them on gel, we will get this pattern. So the enzyme 1 has cut one fragment into 2 pieces, cut it once and generated 2 pieces. So here we have 2 fragments and they are about, one is B is slightly bigger than the A. In case of enzyme 2, it generates 2 fragments also, but one is much bigger than the other. 1 plus 2 will of course generate 3 fragments. It will cut the DNA 2 times. In the other cases, we were cutting DNA. Enzyme 1 and enzyme 2 can cut our sample DNA only once. When we add 1 and 2 together, it is going to cut DNA 2 times and generate 3 fragments, which you can see here. So basically, we can see that the enzymes are cutting at DNA at specific locations. And how can we use this information? So now imagine there's a disease. Here we are looking at, for example, sickle cell anemia. Normal DNA has 3 restriction sites for a particular enzyme, MST2 as it is named here. This is a restriction enzyme, endonuclease. This enzyme can cut this gene, the normal globin gene, into 2 fragments. It will cut it 3 times, once here, and once here. So this whole gene is cut 3 times and 2 fragments are generated from this gene. Here is fragment number 1, fragment number 2. Say there is a mutation and that mutation has removed this particular restriction site. So now what happens? This, our enzyme MST2, will cut it into these 2 pieces and only 1 fragment will be generated of this particular gene. It will not be 2 different fragments. When we run this gel, we will separate these fragments. When initially we saw, as we saw in our previous slide, this fragment will run faster than this fragment. So here, this is a smaller fragment, this is the larger fragment. In case of sickle cell anemia, where the mutation was removed, the whole gene stays together because it is cut at the 2 very ends and the gene is sitting here at a higher location, at a higher level, because it's larger. So this is one way we can detect a mutation. We can use these restriction enzymes to detect a disease. I need to tell you a few more things. If we isolate DNA from a human being and run it on a gel, it is very heavy and it will just accumulate pretty close to the well. It won't move very further because DNA is so large. Generally, when we cut a DNA, with an enzyme, restriction sites are at many places throughout the genome. Here is a gel showing the digested DNA with an enzyme. See how it has made a smear. With PCR, you can see the bands. When you cut the DNA with an enzyme, you don't see the bands, you see the smear. Here also, you will just have a whole smear and you won't be able to see this particular band. When you're using a restriction enzyme to detect a disease, you will not be able to see a band. So how do we see the bands? So we can tell whether we would know whether this gene has lost its restriction site, whether the mutation is there or not. So we will talk about how we can do that, but I will again want to point out that also, like VNTRs, these RFLPs are also inherited in Mendelian fashion. So if you can use this technique to form pedigrees, and you can very easily see which individual in a pedigree is heterozygous, where the person is homozygous, and both with recessive and the dominant form. Here, for example, is another allele that we are looking at. In this allele, there is no restriction site, and here we have a restriction site. So we can use this and build a pedigree, and we can follow this pedigree, this particular gene or mutation in this pedigree. We haven't talked about how we detect the products of restriction endonucleases, whether the DNA has been cut or not. But here I just wanted to show you that these mutations in alleles are also inherited in Mendelian fashion. Next, we will talk about how you can detect DNA restriction endonuclease sites, whether they have been mutated or not.